Hi, this is Mr. McLean. In today's lesson, we're going to be talking about how researchers can study gene expression from different target sequences. So one of the things that we looked at in an earlier video is if we can create what's called complementary DNA, that's going to be one of our more direct ways to understand what genes are actually being expressed. So what we're going to do is we're going to examine some of the different techniques that allow us to study the messenger RNAs that are going to be transcribed from specific genes. So in this section, we're going to take a look at techniques that look for certain patterns of expression coming from specific genes. And also we're going to look at how we can go through there and characterize different groups and see how they're being expressed. So we'll examine a few of those different things. So one of the first things that we want to talk about is the idea of doing what's classified as nucleic acid hybridization. And basically what you're going to need to do is you're going to need to create a probe. So a probe is a artificially generated sequence. It's going to be a single-stranded sequence, and the sequence is going to be complementary to the sequence that you're looking for. So if we knew that a particular gene sequence of interest had a set code, we would want to create something that would be complementary to that particular code so that we can match those things up. And that's going to be the probe. And the probe has a specific fluorescent labeling or tag associated with it so that when it actually matches up, it's going to fluoresce and give a unique color that can be identified by computer-based scanners. Okay, and we're going to take a look at a way in which this can actually be done in some living organisms. So we introduced the term in vitro in an earlier video, which would technically mean in glass. But in some cases, we want to study things in an intact organism. So in situ is going to refer to something that's done in place. So in this particular study, what they were doing is they were looking at embryos of Drosophila. Okay, and it says they used the fluorescent dyes and attached the probes. So they're looking at two specific genes in this particular section here, the WG section and the EN section. And as we go through here, they generated a probe. For the simplicity of this slide, they made one have a fluorescent label of yellow, and then the other one had a fluorescent label of blue. So we knew what the target sequence of the messenger RNA coming from the WG gene would be. So the complementary sequence was generated with that yellow dye. And the same thing was true when we did it with the EN. So we had a different probe that was generated. So when they did this, they're able to go through there, and it talks about as the Drosophila embryo was incubated with this solution of different probes. For in the actual study, they did 5 different probes. So you're going to notice that there's more than just the yellow and blue, but we have some red and green and pink and purple that were associated with that. And what that was going to do is it was going to show you specific areas where we had the expression of that particular gene. So we can go through there and then match it up with the organism as a whole and see that, okay, some of these genes are more specifically located in the head region and some of them are in the abdominal region. So it's a way of doing a whole organism genetic analysis. And obviously we're going to be limited on what organisms we can do that with, but that's what nucleic acid hybridization being done in situ looks like. So one of the techniques that definitely allows us to compare more than one sample and to look at them multiple at a time is what we refer to as reverse transcriptase polymerase chain reaction, or RT-PCR. And a couple things that are going to take place in there is that number one, we have to start with our complementary DNA. And we're also going to use PCR, which we were introduced in an earlier video, which is a way to amplify a particular code. And then we have to run it on gel electrophoresis, which we also showed in an earlier video. So just a quick review of how this works. Complementary DNA is going to be DNA that is generated from only the coding messenger RNA. So they use reverse transcription. and you can review this particular slide here. But what that's going to end up doing is it's going to build our DNA of only the expressed regions. So that's what the complementary DNA or cDNA is going to be. And then we got to duplicate that. So we use PCR, and we talked about how PCR is going to amplify. So you're going to get lots and lots of samples with that. It's going to have the heating and the cooling and the annealing and the expansion component of that. From that point there, they can actually run these on a gel electrophoresis. So in this example right here, what they were doing is they had 6 different embryonic stages that they were studying a given mRNA from. So whenever they took the amplified complementary DNA and ran it on a gel electrophoresis apparatus, what they discovered is that there was no presence of that during the first embryonic stage. You're going to notice it's empty right here. But during stages 2 through 6, there was expression of those genes. So this would reveal some information about how a gene is active at different stages of embryonic development. And here's a little bit of a closer look at that. So they can reveal some of the information from that particular perspective. Okay. Another technique that allows us to look at, in some cases, thousands of genes simultaneously is referred to as a DNA microarray assay. And what that's going to end up doing is it's going to create what becomes almost like a gene chip, kind of from the analogy of a computer-based chip. So they're going to take a tissue sample, whether we're looking at a tumor sample or other samples that we wanted to do comparison against. We're going to analyze and collect the messenger RNA, which is reflective of what's being expressed. We're going to go through there and generate complementary DNA, and we're going to label them, and they're going to have different labels. So they're going to have, the different cDNAs that we're concerned with are going to have different fluorescent labels and different colors. We're going to apply the mixture to a microarray. So basically, it's one of these tiny micro well components, and then it's going to fix the samples of that cDNA onto a glass slide. Okay, and it's going to create a chip that looks something like that. So if we zoom in right here, the idea is that each dot is going to have a little component of the different cDNA that was fused on there. So whenever they run an analysis of this, what we're going to be able to see is that in a particular tissue, which genes were being expressed. So if we're going through there and we might see, an orange color indicates certain genes, a green, yellow, and so forth and so on. So in this case right here, we're noticing that these genes were not expressed in a tissue. So it gives us a little bit of a better understanding if we're trying to look at why certain genes are being expressed in certain cells, and is there an overexpression of something. So this is definitely useful in things like cancer research. Now, another technique that is often used is to try to figure out what would happen if that gene was disabled. Okay, so in that particular case, they're gonna literally want to knock out a gene or use what's referred to as an in vitro mutagenesis, which is basically to alter or destroy a particular gene function and then try to put it back into the organism to see what the impact is going to be. Now, there's a couple different ways in which that can be done. And obviously, this is not something that we're going to be able to use on humans, but we can do it on different organisms to try to get a better understanding. So probably the most powerful tool right now for being able to go through there and make specific gene edit cuts is to use the CRISPR-Cas9 system. And just to give you a little bit of background as far as how this system operates, CRISPR-Cas9 is derived from a immune system capability found inside bacterial organisms, which allows them to essentially cut up and destroy foreign DNA. So if you have a virus that were to inject a DNA sample into them, they'd be able to make specific cuts and basically render that genetic material not useful. So in part, when we take a look at what's involved with the CRISPR system, the first thing that we have is called the Cas9 protein. Okay, and the Cas9 protein has sites that allow it to cut DNA, but the Cas9 protein is under the direction of what's called guide RNA. So guide RNA fits inside this Cas9 protein, kind of like this picture right here. And what that's going to do is it's going to guide it to a particular sequence that's going to be complementary. So in a natural system, you know, part of the immune capability is that there are different guide RNAs that are going to target things that are abnormal. From an engineering standpoint, the biologists are able to generate a guide RNA that's complementary to what we're targeting. So if we're looking for a particular target, we can generate a guide RNA sequence, which is going to have a complementary component to it, and it's going to be able to seek out and basically target a particular area that we're focusing on. So what we're going to find is that the Cas9 is going to have its active capability, and it's going to match up. So it says in the nucleus, the complementary sequence of the guide RNA is going to bind to the target gene, and the active site of the Cas9 is going to line up, and it's going to cut the DNA at that particular target. So when we do that, what that's done is it's more or less disabled. It's created a break within that target DNA. So that's one way in which we can kind of disable. And then obviously, if we have the enzymes and the necessary components, we can allow this to reheal itself. And from a gene therapy standpoint, we can incorporate a repair gene into that location. Okay, so if you take a look down here, here's our two options. It says scientists can disable, which is knocking out the target gene, so they can study what it functions like if it's not there. So that would be one way to do that, knockout, as it's referred to. In the other case there, we can allow it to be repaired, and if we have the corrected gene available, this is how we want to try to utilize gene therapy. Okay, and we'll talk more about that in a later video there. We also have the capability of interfering using RNA interference, and we had talked about that in chapter 18, which basically means that we can block a gene from being able to, be able to go through there and generate protein. All right, so we can't mutate humans, clearly. So what we rely on are GWASs, which are genome-wide association studies. And what we're looking for are called single nucleotide polymorphisms, or SNPs. And a SNP is something that we can utilize our PCR technology and expansion technology. But the idea is that when you look at a population of people that have a similar genetic outcome, what we do is we look for something that's similar. So we're trying to find out, in a hypothetical scenario, we've got a population of people that are normal for a particular ailment, so they don't display the phenotype. But we have a group which was analyzed. All of these individuals have the disease-causing allele. And one of the things that is revealing when we look at them a little bit more closely is that the normal population had a T at this position, and the population that suffers from the disease has a C. So once we've identified that, what we can actually do is we can use that as a genetic marker, and that can be used for diagnostic recognition of that particular allele. So hopefully that was helpful, and we'll continue to build on this chapter as we move through.